Proceed. Wait करें या proceed करें? 9:30 तो बज गए. 2015. Okay, so we work on couple of different aspects. First of the thing is which I'm going to present today is about drug resistance. Drug resistance in Plasmodium falciparum. Now, well, okay, you are aware about malaria, and what's happening is malaria. Initially, chloroquine was used to treat malaria. Okay, but what's happening is in 2004, chloroquines. Well, okay. Subsequently, they started not working. Yeah, please come in. It was failing. The parasite was becoming resistant to chloroquine. So 2004, WHO recommended the use of artemisinin. Now, artemisinin is a traditional Chinese herb. Okay, Chinese people used to drink that artemisinin tea. Okay, now gradually the product was identified. It was isolated, and it was termed as artemisinin. Artemisinin is very potent. You add artemisinin to nanomolar amount, and it kills the parasite. So it's a good drug. Okay, so people suffering from infection, you give artemisinin. Yeah, please come in. We're just giving an overview. We'll start. We'll cover all this in the slides. Okay, so they gave artemisinin. It killed the parasite. So that's great. But remember one thing. This is a smart parasite. It has been persistent from the time of pharaohs in Egypt. In fact, King Tutankhamun, okay, his body, the coffin, his body was extensively analyzed, and they found traces of malaria parasite. So the parasite has been persistent since that time also. It's still persistent. So people use artemisinin to treat malaria. Well, okay, good, all good. But, the problem with developing and underdeveloped countries is you get spurious drugs. Okay, you're buying artemisinin from the chemist. Okay, but what will happen is it might not contain the appropriate dose of artemisinin. So if that's the scenario, what's happening is you give artemisinin, you have malaria, you take artemisinin. Artemisinin will kill 99 parasites. That one parasite is still alive. Okay, and the problem is artemisinin has a lower half-life. That means whatever dose you give, after 2 hours, it becomes 50%. After 2 hours, again 50%. So the parasite, that one parasite that survived artemisinin, okay, after 2 hours, the dose comes low. So that parasite has now seen artemisinin. So it can survive. That will multiply. So artemisinin resistance is emerging. Not in India, yet not in India. But it's in Thailand, Myanmar, Cambodia, it's there. So it's just a matter of time it's going to come to India. Okay? So, should we still wait or should we? Let's start. Okay. So what I'm going to talk about is, there are three different terms here. One is heme. Okay? Heme is an important factor. Keep this in mind. Okay? Heme. And the second one is artemisinin, which short form we write as ART. And the third one that we are going to talk about is a protein called as K13. So these are the three important factors that we are going to talk about in this presentation. Okay, now, moving on. I'm sure you are aware about life cycle of malaria parasite. Infection starts when an infected female Anopheline mosquito, they inject sporozoites. These are motile form of parasites in the bloodstream. Once injected, within 30 minutes, they migrate to the liver. So that's the mandatory first step. They migrate to the liver, infect hepatocytes. You still don't get any symptoms of malaria. There is no fever, there is no body ache, there is no chill. So you don't detect malaria parasites at that stage. Within the liver, they multiply within hepatocytes. Ultimately, the hepatocytes lies. Not only the parasites multiply, they differentiate also. They differentiate from sporozoite to a form called as merozoites. The hepatocytes lyse and liberate merozoites into the bloodstream. So in the bloodstream, they infect red blood cells. This is the time you start seeing symptoms of malaria. In the red blood cell, they progress from a ring stage, it looks like a ring. Ring stage is, you've done bacterial growth curve. That's the lag phase, basically when it is adapting. The parasite has infected a red blood cell. What is a red blood cell? Red blood cell, there is no nucleus, it's just a bag of hemoglobin. There is no nucleus, it cannot multiply. It's a terminally differentiated cell. So within which the parasite, it has moved into an empty room. You have moved into a new apartment. Okay, now you have to make it its home. You have to get the furniture, you have to get cooking gas, microwave, everything. So the red blood cell doesn't do anything. I'm inert. You have come within me, you stay there, but make this your home. So the parasite modifies the red blood cell. It takes hemoglobin, digests hemoglobin, so that what happens is it gets nutrients from it. Not only nutrients, hemoglobin what? It's heme and globin. So what you get is, globin is a protein, you digest, you get the amino acids. What about fatty acids and all? So the parasite, what happens is it modifies the red blood cell so that it can take fatty acids, it can take small amino acids that are deficient in hemoglobin, okay, so as to make it its home. Okay. So from ring stage you go to trophozoite stage, where the main hemoglobin digestion happens. Okay, to get the nutrients, the parasite is now thriving. From a lag phase, it has gone into exponential phase. Okay, it is thriving, it is digesting hemoglobin. The problem with hemoglobin is, once you digest hemoglobin, you generate heme. Now, heme generates reactive oxygen species, which is bad for the parasite. So it's an oxidative stress that is generated. You need nutrients from hemoglobin, but you also have to take care of the free heme that is there. Because too much free heme will generate an oxidative stress. That comes the heme factor, the oxidative stress. So if there is too much oxidative stress, it's bad for the parasite. The parasite will die. So what the parasite does is, the parasite, that's the parasite, takes in hemoglobin, okay? It digests hemoglobin, and most of the free heme is polymerized into a crystal called as hemozoin. This is what you say as malaria pigment, okay? This is a black pigment that is inert hemoglobin or hemozoin. Okay. So it has taken care of, it has digested hemoglobin, okay? It has taken care of free heme by polymerizing it into hemozoin. But a living system is a complex system. Unlike physics, where there is 0 and 1, it happens or it doesn't happen. But living system is complex, it's never one way or the other. None of the process is 100% efficient. So there is still some free heme. That creates a problem, and we are going to talk about that as we go further. Okay, so overall, the point is there is a lot of heme generated in the parasite because of hemoglobin digestion. This heme is bad. Okay. Now, moving on to the next slide. Okay. Now comes the second factor, artemisinin. Artemisinin came from Chinese herb, okay? 1972, UU2 discovered artemisinin. She purified it and showed that it has this antimalarial property. Okay, she got a Nobel Prize for artemisinin discovery. Now, artemisinin, this is what it looks like. Now, the thing to remember is artemisinin is inactive. When you take that drug, it is a pro-drug. So from inactive state, it has to be converted into an active state. Okay, who does it? It is done by heme. Artemisinin, if you see, there is an endoperoxide bond here. Okay. At this stage, if the endoperoxide bond is linked it is inactive. Heme cleaves that endoperoxide bond to activate artemisinin. Okay, so heme plus artemisinin is a lethal combination. You just give artemisinin, if there is no heme, there is no activation. Okay, in presence of heme, artemisinin gets activated. So, point to remember is heme is essential for activation of artemisinin. Right? Okay, what does it do? Heme reacts with artemisinin, iron protoporphyrin. This is what exactly is the chemical name of heme. Okay, so it reacts with artemisinin and forms free oxygen radicals. These are reactive oxygen species. So, once heme activates artemisinin, it leads to production of reactive oxygen species, which are stress for the parasite. That's what kills the parasite. There is formation of reactive oxygen species which target protein, lipids, and DNA, causing a total chaos in the parasite. So that, it's killing at all different aspects. Unlike, where you have an inhibitor against an enzyme, where it will inhibit that enzyme only. But artemisinin acts on DNA, lipids, and proteins. So there is damage at all front, and the parasite dies. Not only that, what artemisinin also does is, remember, majority of the heme is polymerized into hemozoin. Artemisinin prevents hemozoin polymerization. So free heme is there. Free heme generates reactive oxygen species, killing the parasite. So it's a double-edged sword. Not only it damages protein lipids, it also prevents hemozoin polymerization. And it kills the parasite. Great. Artemisinin is an excellent drug, a miracle drug. Okay, but it's a smart parasite. The parasite has thrived evolution. Okay, you'd be surprised to know malaria parasites are found in all different species. You get ducks get malaria, reptiles get malaria, mice gets malaria. Fishes don't get malaria. Why?
They have not
Fishes, red blood cells, if you cut a fish, do you see? Okay, but they don't get malaria. Why not? It's a simple logic. It's not scientific. It's just simple. They survive in water. So where is the mosquito going to bite the fish? It's in water. So mosquito cannot go underwater and bite the fish. Okay, but reptiles, even if they have hard skin, they get malaria. Monkeys, of course they have malaria. A monkey malaria parasite now has been seen in human also. It can infect humans also. Okay, so basically that's what is happening. As we are encroaching their habitat, we are getting their pathogens also. You've heard about COVID, Ebola. These are coming into humans. So same thing, one of the malaria parasite that infect monkeys is coming into humans also. Okay, so basically artemisinin and heme is a lethal combination, and artemisinin is a miracle drug. But a smart parasite generates resistance to artemisinin. So this is where you see the red shade portion is the artemisinin resistant, blue one is the artemisinin sensitive. It came up in Thailand-Cambodia border, started here, okay, and then gradually spread. Bangladesh, there are few cases, 1 or 2. Now, those cannot be confirmed because these days, you travel from one country to another. Whether the parasite came from Thailand, Cambodia with a person or not, you don't know. But it's not widespread. So see, this is blue. There's no red, okay? But in these areas, Thailand, Cambodia, Myanmar, there is a lot of red. These are artemisinin resistant parasites. Those are thriving there, okay? And India is right here. It's very likely that by this time, there are some cases of artemisinin resistance in India also. And that's bad because there is no other antimalarial drug currently in the pipeline that will kill artemisinin resistant parasites. So if you have artemisinin resistant parasites, it's bad. Okay? Now, what? Okay, fine, I have artemisinin resistant parasites in me. I take more artemisinin so that now the parasites are exposed to higher artemisinin concentration, they will die. Agreed? That's a good point. The drawback, one of the drawback is artemisinin is toxic. You cannot take high dose, like, paracetamol. If you're sick, you take paracetamol. But you cannot take paracetamol every hour. There's a doctor recommended dose. If you take paracetamol every hour, your liver is going to get damaged. So every 6 hours, 1 paracetamol is the maximum you can take. So in a day, maximum 4 paracetamols you can take. Same case with artemisinin. You cannot just, take increased dose and believe that it's going to kill. Okay, so basically resistance, the parasite that becomes resistant, they have delayed response to artemisinin. They don't die in artemisinin as fast as sensitive parasite. They multiply, they make gametocyte, spread the infection. Okay, again you use more drug, again it's developing resistance. So as you are taking more and more and more of drug, this is toxicity factor and you're breeding resistance. Okay, so you have to be careful and you have to understand what exactly is the mechanism of resistance. Then only you can target those resistant parasites. Okay. Till now, we are in this good page. Okay. Heme is important for artemisinin activation, and artemisinin is a miracle drug. They cannot function independently. They both need to be present for the drug to have antimalarial property. Right. Now, what exactly is artemisinin resistance? Now, in 2014, okay, just before I joined here, there was a paper published that, there is a parasite protein which confers resistance to artemisinin. Okay, how do you do that? You have a parasite which is sensitive to artemisinin, and you have a parasite that is resistant to artemisinin. Have you heard of this term, whole genome sequencing? When you take the whole genome, you sequence, and then you align side by side to find out which genes have developed mutation. Those will likely confer towards resistance. If those mutations are present in the resistant parasites, they will confer resistance. Great, good idea, do it. There are around 800 genes that have mutations. Now there is a problem. 800, which one came first? Okay, a protein, if it is mutated, that mutated protein will affect its binding partner also. So these two normally interact. This has developed mutation. In order for them to interact, this will also develop mutation so that they can interact. But you don't know which is the first. So among 800, which came first? No lab can work on 800 proteins. How are you going to work on 800 proteins? It's not easy. So what they did was they treated the parasite with low dose of artemisinin subsequently step by step. And what they found out was there is a protein called as KELCH13 or K13. Okay, the full form is KELCH. Short form is K13, where mutation confers artemisinin resistance. Okay, so KELCH13, it's a big protein. Okay, it's a huge protein of 726 amino acids. And what people have generally seen is the mutations are conferred in the beta propylar region. This looks like the beta propylar region. It's a propeller of a blade. The mutations, all those marked here, these mutations confer artemisinin resistance. So that brings KELCH13 into focus. This is the protein that confers artemisinin resistance. Okay. So now, how does it do it? How does it confer artemisinin resistance? A lot of studies have been done. Okay, some people say that, parasites with KELCH13 mutation, the transcription profile is different. Okay, transcription profile different means, there are 300, 400 genes that are upregulated, 300, 400 genes that are downregulated. But same issue, 800 mutations and so many genes up and down. You don't know what is causing that. Okay. The second thing is these cells, artemisinin-resistant parasite, can adapt more to oxidative stress. So that's why they are thriving. That's also an indirect relation. Okay, so nothing is known directly. What was postulated is, remember, this is the parasite. KELCH13 is localized here, at the tip of the parasite. It is involved in hemoglobin uptake. Okay, keep this in mind. Okay, it is involved in hemoglobin uptake. Hemoglobin is taken in, digested to form heme, which activates artemisinin. Now, mutant KELCH13 takes less hemoglobin. So if there is less hemoglobin, there is less heme. If there is less heme, there is less activation of artemisinin. So that's how the parasite survives. Okay? So now there is a connection between all these three. Mutation takes less hemoglobin, there is less heme. Less heme causes less activation of artemisinin. Okay? So this is how reduced, it is located in parasite periphery. Mutation leads to lower amount of KELCH13. Okay, even if there is low amount of KELCH13, that will also cause less hemoglobin uptake. Okay, less heme formation, less activation of artemisinin. So that's how the parasite survives. But these all studies, till date, they indirectly implicate KELCH13 for artemisinin resistance. You have to find out how exactly it does. So you start with an easy question. Okay? If KELCH13 is involved in hemoglobin uptake, okay, generates heme, first question is, does it bind heme? Okay, so now don't look into complexity, just consider there are 4 proteins here. This is the wild type protein, KELCH13, which is sensitive. It is present in sensitive parasites. And these 2 mutants are present in the artemisinin resistant parasites. These 2 mutations confer artemisinin resistance. The 4th one, this one, it is a mutation in KELCH13 but does not confer resistance. Okay, so we have all these 4. Now, you look into the protein, the red spot is where the heme binds KELCH13. KELCH13 binds heme. Okay, so now you just follow this dotted line. Okay, 1 and 4 are the sensitive ones, artemisinin sensitive ones. 2 and 3 are the resistance ones. Now, if you look into the red dots, what you see here is the red spots are mostly below the dotted line for the artemisinin sensitive ones. But in artemisinin resistant ones, the red spots are mostly above the dotted line. And this is where the propeller region is. This is where the mutation confers artemisinin resistance. Okay, so it seems that because of the mutation, there is a shift in heme binding region in KELCH13. Okay, what are the blue spots? The blue spots are artemisinin. For artemisinin activation, you need heme. So heme is bound, and wherever heme is bound, artemisinin docks. There is no blue spot where there is no red spot. Okay, so wild type, the sensitive parasites, they have heme bound lower the dotted line, while resistant parasite, the heme binding sites are different from the sensitive parasites. So there is a difference in heme binding. How will it affect? The difference in heme binding, how will it affect artemisinin? That we show in the next slide. This heme, if you measure absorbance of heme has a peak around 410 nanometer that is called as a Soret peak. Okay, that is characteristic of heme. If a protein binds heme, protein has what absorbance? When you measure a protein concentration, you measure absorbance. What nanometer do you measure? 280 nanometer. Now, why 280 nanometer?
Because there is some aromatic.
Okay. It absorbs at 280 nanometer. The aromatic amino acid, tryptophan, tyrosine, and phenylalanine, they absorb. Okay. Same case, heme has around 410 nanometer peak called as Soret peak. So, a protein by itself will not have 410 nanometer peak. If it has bound heme, it will show that 410 nanometer peak. So that is what is happening is, if you see the blue, just the dotted line, the protein does not have any 410 nanometer peak. You add heme to it, it binds heme, it is showing the blue line, there is a 410 nanometer peak. The peak is slightly shifted because of heme binding. But this peak, the blue line peak, is characteristic that the protein binds heme. Now, look at the blue line across 1, 2, 3, 4. If you compare all the 4, what you are seeing is the blue line is much more prominent in 1, 2, but it's there, but it is behind the red line. That's why you don't see it. But if you see these two, it is less. Okay. What does it mean? The mutant, the artemisinin resistant parasites, which has Kelch mutation, they bind less heme. Because higher the heme binding, the greater will be the peak. The blue peaks are much more prominent in this and this. This is the wild type Kelch, and this is a Kelch mutant that does not confer artemisinin resistance. So, Kelch binds heme. Okay? Mutant Kelch, which confers artemisinin resistance, they do not bind heme as efficiently. Okay? What does it do? Kelch has bound heme. Heme is required for artemisinin activation. Right? Now, when you add artemisinin to the protein that has bound heme, what you see is the blue peak has now gone up to the red peak. The red peak shows artemisinin activation. Wild type parasite, you see the blue has shifted above. Even in this Kelch mutant, which is artemisinin sensitive, it has also shifted above. So these two parasites activate artemisinin. That's why they die. But resistant parasites, they bind less heme, they cause less artemisinin activation. You see, there is barely any shift from blue to red here. Same case here. So that is how the resistant parasite survive artemisinin treatment. Okay, now these 4 we have seen. What about these 2? These 2 are merely showing that, any protein that binds heme does not activate artemisinin. It has to be Kelch 13 to activate artemisinin. These 2 proteins are known malaria protein. This is BSA. BSA, bovine serum albumin. It is a known heme binding protein. But you see the red to blue shift? There's barely anything. The red peak is very prominent here. It binds heme, but does not activate artemisinin. Same case, you see the red peak is very prominent in another parasite protein that binds heme, but the blue peak is barely going up. So that's the unique thing about Kelch-13, that it binds heme, that bound heme activates artemisinin. Any protein which binds heme does not lead to artemisinin activation. You need Kelch-13 for artemisinin activation. And that's how the parasite is smart, that, I cannot control hemoglobin uptake. Okay, what I can control is artemisinin activation. So what I'll do is, I'll mutate Kelch-13 protein so that it binds less heme, I'll lead to less artemisinin activation. That's how I will survive. Okay, so that's how the smart parasite is. Okay, so this is just the graphical representation of that, showing that the wild type and the mutant, they activate artemisinin very efficiently compared to the resistant parasite with mutated Kelch-13. Are we clear in all this? If you have any doubt, stop me. But the problem still persists. What exactly is Kelch-13? It's not an enzyme. You cannot do an assay. You have done alkaline phosphatase assay. Have you done in MSc practical or something? You isolate an enzyme called alkaline phosphatase, and then after that you check its activity. Okay, PNPP you have heard? No. Okay, so how do you check its activity? You take a substrate, add enzyme. If the substrate is converted to the product, you know the enzyme is active. Okay, PNPP, paranitrophenol phosphate, is converted into paranitrophenol, which is yellow in color. So the more yellow color you see, the more active the enzyme is. But Kelch is not an enzyme. So there is no assay you can do to check enzyme activity. So that remained in 2014 to 2024. It remained a puzzle. What is Kelch-13? How do you know that, a mutation in Kelch-13, what does it do? What does it cause? Okay, so a clue came that was even identified in 2014 only, that Kelch-13 is very similar to a human protein called as KEAP1. But, you have, sometimes what happens is, if you read a scientific paper, you'll find that a discovery, once the protein was discovered, let's say 10 years ago, they would put, this protein is very similar to another protein. After that, you come across each paper, just because that paper has said that this is similar to that protein, everybody puts that line in. Nobody is willing to check. So we started with, we start with a simple problem. Okay, for 10 years, people have been saying Kelch-13 is similar to KEAP1 in terms of sequence. Okay, but have anybody looked into it? If it is similar in sequence, is it similar in function? So that's the idea we started with. That what is the function of Kelch-13? How can we check it? What we know is Kelch-13 is very similar to a human protein called as KEAP1. Okay? There's a problem in malaria. You cannot modify the red blood cells. There's no protein synthesis. There's no lipid synthesis. There is no nucleus even. So how can you make a transgenic erythrocyte? You cannot make that. You cannot even modify it. Okay, it's a bag of hemoglobin. But KEAP1, it's a mammalian protein. It's a human protein. Okay, it is present in human cells. And human cells, you can modify. Okay, so we started with the idea that Kelch-13 has been said similar to KEAP1, right? Is it sequence similar, or can it also do this function similar as KEAP1? How do we check it? We have to first know the function of KEAP1, okay, to check. Be with me. If you don't understand anything, stop me right and there. I'll go over it 10 times. Okay, what does KEAP1 do? Okay, this is a mammalian cell or a human cell. Okay, it has KEAP1 protein, right? This is the KEAP1 protein. KEAP1 protein binds a transcriptional factor called as NRF2. You know what a transcription factor is? It goes to the nucleus, binds the promoter region, okay, and it activates transcription. Okay. So what does KEAP1 do? KEAP1 binds the transcription factor, it ubiquitinates and degrades the transcription factor. If there is degradation of the transcription factor, there is nothing to go to the nucleus, nothing to trigger transcription. Okay, that happens in normal condition. But if there is reactive oxygen species or stress in the cell, what KEAP1 does is the conformation of KEAP1 changes. If it changes, it can no longer bind NRF2, no longer degrade NRF2. NRF2 is now free. This NRF2 goes to the nucleus and it expresses stress response genes. So there is stress because of reactive oxygen species. NRF2 is released, NRF2 migrates to the nucleus, expresses stress response genes. These stress response genes take care of the reactive oxygen species or oxidative stress. That is how human cells function. And that is how KEAP1 functions. Now you know the function of KEAP1. KELS13 has been said to be similar to KEAP1. You can check that. How can you check it? You remove KEAP1, put KELS13 in. Does it do the same thing? Okay. So, this is what is showing KELS13 and KEAP1. It's very similar, especially in the propeller region, it is very similar to the KEAP1. And mutations conferring artemisinin resistance are there in this region. Okay, so this is basically KEAP1. Now you know the function of KEAP1. KEAP1 degrades NRF2, which is a transcriptional factor. Okay, as a result, there is no stress response genes expression. Okay. So, we took a cell line, a human cell line. This is called as DU145. The advantage, DU145 cells have low KEAP1. So, you can put KELS13 in there and see if it compensates for low KEAP1 function, right? So these are human cell line deficient in KEAP1. So you can express K13 with minimum interference from KEAP1 because there is as such low KEAP1 in these cells. Okay, so now you take these cells, you express KELS13 in these cells. Okay, so all these studies in the lab, it was done by her. Dr. Smriti Khanna, she's a postdoc in the lab. Okay, so now you see, an image conveys much more than 100 lines. An image you remember, lines you won't remember. Okay, so you see these cells. These are the cells where you express KEAP1. In these cells, there is low KEAP1. You have overexpressed KEAP1, you have overexpressed KELS13. This is as a GFP fusion so that you can see where it is. And for every experiment, remember one thing, you need controls. Without controls, your experiment doesn't have any meaning. Okay? If you're doing an enzyme assay, you have to take a control where there is no enzyme. You have to take a control where there is no substrate. You have to take a control where there is no ATP, just to make sure there is no background signal you are detecting. Okay? So KELS13 is fused to GFP so that you can check where exactly is KELS13, and you have to take just GFP as a control. In all of these, what you see, the green fluorescence is, this is the cell, this is the cytoplasm, and blue is the nucleus, right? You see the signal, all the proteins, keep one cytoplasmic, Kelch13 cytoplasmic, GFP cytoplasmic. So all the proteins are there where they need to be. Cytoplasmic protein, these are present in the cytoplasm. Okay? Not only you have to do imaging, you have to also do Western blot also to confirm, the fluorescence that you are detecting is because of your protein. Okay? So this Western blot was done. This is the Kelch13 GFP band, this is the GFP band. Okay? This is with another antibody, and this, what is GAPDH? It's a loading control. This signal you're getting, maybe it's possible you did not load sample here. So you need a loading control to show that I have samples in both the lanes, but we are detecting Kelch13 only in the transfected cells that are expressing Kelch13. So you have to confirm by Western blotting. And this is where you have also overexpressed KEAP1. You have to compare Kelch13 with KEAP1. So if you have overexpressed Kelch13, you have to overexpress KEAP1 also. This is the control you have to do. Okay, so this is also expressing. Native cells have very low KEAP1, and you need what? You need loading control. So all the proteins are expressing. You now have an handle to check. Moving on. There came the surprise for you. That's the thing. Science is interesting, it's difficult, but you try to answer one question, 10 more questions come. That's the good thing and bad thing about science, depending upon how you take it. Right. So, now if you see here, what is this thing? For roughly looking at it, what you're seeing is the intensity of the band is increasing, right? While this band is more or less same in all. What does it mean? It means that what's happening is, you remember those cells where Kelch13 is overexpressing the green fluorescence? You culture those cells in low heme, a media that has low heme, and gradually you increase the heme concentration. Okay? The cells are cultured in a media. You have heard about RPMI media? Okay? So RPMI media, and you have heard about FBS, fetal bovine serum. Cells are cultured in RPMI media plus fetal bovine serum, which gives lipids and other nutrients. Fetal bovine serum has heme. Okay? Because it is a serum, it has heme. What you can do is, you can remove heme from the serum. Okay? So you remove heme from the serum and add little bit gradually increasing amount of heme. What we saw was, as the heme amount is increasing, you see this is 0, no heme, 0.01 micromolar till 5 micromolar. As heme concentration is improving or increasing, the intensity of Kelch13 is increasing. Okay? And you can even see in the fluorescence image also. You just look at this magnified image. This is in the normal media, this is in the heme minus media. You see the green staining intensity? It's much low. But when you add heme to it, the green staining intensity is coming back. Okay? So it seems that, heme is providing a stability for Kelch13. Kelch13 binds heme. Kelch13 binds heme. Heme provides a stability for Kelch13. Okay? Now, what is heme? So as heme concentration is increasing, Kelch13 is becoming much more stable. Okay? What is heme? Heme is iron protoporphyrin. Okay? Have you read about heme biosynthesis pathway? You have read about it? Now, starts with aminolevulinic, okay, aminolevulinic synthase, okay, uroporphyrinogen, coproporphyrinogen, protoporphyrinogen, protoporphyrin, ferrochelatase adds the iron to it. You'll read it. Okay, you'll read it in biochemistry. Okay, so now heme confers stability to Calc13, right? But heme has, what is heme? Heme has Fe protoporphyrin 9. This is what heme is. Okay, so what is required to confer stability in heme? Is it iron? Is iron enough, or is protoporphyrin enough to confer Calc13 stability? How you can check it? You can check with heme analogues which does not have iron. Instead of iron, it has zinc. So that will tell you what is the importance of iron. You can even check with just protoporphyrin, right? So what is happening is you have protoporphyrin and you have zinc protoporphyrin. You see the level of Calc13, it's low, whereas in heme it is high. So you need both iron and protoporphyrin to confer stability to Calc13. Just protoporphyrin is not enough. You also need iron to it. Okay? So only heme and not heme analogues stabilizes Calc13. You need both of them to stabilize Calc13. Okay, that also puts an important factor. Calc13 binds heme. Okay, binding heme stabilizes Calc13. Okay, and you need the full heme molecule. Partial heme molecule won't be sufficient. Right? That's another thing also that is very important. Stabilizing a protein. When does a protein become stable? A protein becomes stable if it is resistant to protease digestion. Right? Okay, a protein, if it is unstable, protease is going to cut and digest the protein. If it is stable, the protease is not going to digest the protein. Okay, so now focus on this SDS page. Focus on these blue bands. Okay, what are these? These are protein digestion products. If a protein is there, it is digested, you see low molecular weight products. Okay, so this is the Calc13 protein. You add trypsin to it, the protein is digested. You take Calc13 protein, bind heme to it, add trypsin. Do you see those products? No. So that's what the heme does to Calc13. It binds to Calc13, provides stability and resistant to protease digestion. Okay. Same scenario here also. You need the full heme molecule. If you have just protoporphyrin, if you have zinc protoporphyrin, if you have just protoporphyrin, you still get this degradation product. Zinc protoporphyrin, you still get degradation product. Iron, it is iron protoporphyrin. Heme is iron protoporphyrin. Just iron is not able to protect it. So you need the full heme molecule to stabilize Calc13. So that's how heme protects Calc13. You need full length Calc13 stabilized by heme. Right? Okay. Now, okay, Calc13 is stabilized. Function. What is the function of Calc13? Okay, you remember that KEAP1 binds NRF2. It degrades NRF2. Transcription factor, it degrades NRF2. Right? So, now, this is what is happening is that now when you overexpress Calc13, focus on this part. This is cell without Kelch-13, just GFP, this is cell with Kelch-13. What is the NRF2 level in the cell with Kelch-13? It is lower. That kind of matches here. Keep 1 binds NRF2, degrades NRF2. Instead of keep 1, if you have Kelch-13, it will bind NRF2 and degrade NRF2. Whether it binds or not, we don't know yet. But what we see here is it degrades NRF2. Same as keep 1. Okay, now we have a functional assay to check Kelch-13. It degrades NRF2, so that scenario is good. The question is, does it bind NRF2? It cannot degrade unless it binds NRF2. So this is what is happening is you gradually increase the heme concentration. This is what Kelch-13 is doing. Kelch-13, it binds NRF2. Kelch-13 binds NRF2, right? But as heme concentration is increasing, heme binds Kelch-13. As heme concentration is increasing, what is happening to the bound NRF2? It is going down. It seems, the scenario is very good that Kelch-13 binds NRF2. It degrades NRF2. But as this heme molecule is coming, a heme molecule is binding Kelch-13, NRF2 is getting released from Kelch-13. As gradual increasing amount of heme is binding, lower amount of NRF2 is getting released. So heme displaces NRF2 from Kelch-13, right? That's the first scenario. The second scenario is this one. If heme displaces NRF2, the NRF2 is now free. NRF2 is not degraded. So NRF2 level is increasing. From this point to this point, NRF2 level is increasing. That's the second proof that in presence of heme, K13, in absence of heme, K13 binds heme, degrades NRF2. When heme comes in, NRF2 is released, no longer degraded, level accumulates. That's the second scenario. The third scenario is this one, where you actually make the protein. You actually express the protein in bacteria and you see whether the same thing is happening or not. As you can see here, in presence of heme, there is less K13 bound to NRF2. If you compare the intensity of these two bands, this is lower in presence of heme compared to absence of heme. So heme prevents NRF2 from associating with Kelch-13. Okay? What is heme? Heme generates reactive oxygen species. Heme is bad. Okay? So how do you measure? You see, the more the green intensity, the more is the reactive oxygen species. So you have two cell lines. One is expressing Kelch-13 and the one is expressing GFP. So if you treat with heme, you see the intensity of the green fluorescence here. It's so high compared to absence of heme. Okay? But this is seen only in the when Kelch-13 is present. When GFP is present, you see the green intensity has barely gone up. Okay? So now what is Kelch-13 doing? Heme generates reactive oxygen species, but you need 100 micromolar heme to generate reactive oxygen species. If Kelch-13 is there, 5 micromolar heme is sufficient to generate reactive oxygen species. So that's what Kelch-13 does. It generates reactive oxygen species, okay, so that at a lower heme concentration. Okay? So in fact, it stimulates 4 to 10 times more reactive oxygen species as compared to only heme. A Kelch-13 bound heme generates ROS much more efficiently than just free heme. Okay, that is the function of KELS13, to generate reactive oxygen species when heme is coming. Okay. What is happening to NRF2? If KELS13 releases NRF2, what is NRF2? It's a transcription factor. Under stress, what would the transcription factor do?
It will come to localize into the nucleus.
It will go to in the nucleus because it needs to express stress response genes. And not heme analogs stabilizes KELS13. You need both of them to stabilize KELS13. Okay, that also puts an important factor. KELS13 binds heme. Okay, binding heme stabilizes KELS13. Okay, and you need the full heme molecule. Partial heme molecule won't be sufficient. Right? That's another thing also that is very important. Stabilizing a protein. When does a protein become stable? A protein becomes stable if it is resistant to protease digestion. Right? Okay, a protein, if it is unstable, protease is going to cut and digest the protein. If it is stable, the protease is not going to digest the protein. Okay, so now focus on this SDS page. Focus on these blue bands. Okay, what are these? These are protein digestion products. If a protein is there, it is digested, you see low molecular weight products. Okay, so this is the KELS13 protein. You add trypsin to it, the protein is digested. You take KELS13 protein, bind heme to it, add trypsin. Do you see those products? No. So that's what the heme does to KELS13. It binds to KELS13, provides stability and resistant to protease digestion. Okay. Same scenario here also. You need the full heme molecule. If you have just protoporphyrin, if you have zinc protoporphyrin, if you have just protoporphyrin, you still get this degradation product. Zinc protoporphyrin, you still get degradation product. Iron, it is iron protoporphyrin. Heme is iron protoporphyrin. Just iron is not able to protect it. So you need the full heme molecule to stabilize KELS13. So that's how heme protects KELS13. You need full length KELS13 stabilized by heme, right? Okay. Now, okay, KELS13 is stabilized. Function. What is the function of KELS13? Okay, you remember that KEAP1 binds NRF2. It degrades NRF2. Transcription factor, it degrades NRF2, right? So, now, this is what is happening is that now when you overexpress KELS13, focus on this part. This is cell without KELS13, just GFP, this is cell with KELS13. What is the NRF2 level in the cell with KELS13? It is lower. That kind of matches here. KEAP1 binds NRF2, degrades NRF2. Instead of KEAP1, if you have KELS13, it will bind NRF2 and degrade NRF2. Whether it binds or not, we don't know yet. But what we see here is it degrades NRF2. Same as KEAP1. Okay, now we have a functional assay to check KELS13. It degrades NRF2, so that scenario is good. The question is, does it bind NRF2? It cannot degrade unless it binds NRF2. So this is what is happening is you gradually increase the heme concentration. This is what KELS13 is doing. KELS13, it binds NRF2. KELS13 binds NRF2, right? But as heme concentration is increasing, heme binds Kel-13. As heme concentration is increasing, what is happening to the bound NRF2? It is going down. It seems, the scenario is very good that Kel-13 binds NRF2. It degrades NRF2. But as this heme molecule is coming, a heme molecule is binding Kel-13, NRF2 is getting released from Kel-13. As gradual increasing amount of heme is binding, lower amount of NRF2 is getting released. So, heme displaces NRF2 from Kel-13, right? That's the first scenario. The second scenario is this one. If heme displaces NRF2, the NRF2 is now free, NRF2 is not degraded. So, NRF2 level is increasing. From this point to this point, NRF2 level is increasing. That's the second proof that in presence of heme, K13, in absence of heme, K13 binds heme, degrades NRF2. When heme comes in, NRF2 is released, no longer degraded, level accumulates. That's the second scenario. The third scenario is this one, where you actually make the protein. You actually express the protein in bacteria and you see whether the same thing is happening or not. As you can see here, in presence of heme, there is less K13 bound to NRF2. If you compare the intensity of these two bands, this is lower in presence of heme compared to absence of heme. So heme prevents NRF2 from associating with Kel-13. Okay? What is heme? Heme generates reactive oxygen species. Heme is bad. Okay? So how do you measure? You see, the more the green intensity, the more is the reactive oxygen species. So you have two cell lines. One is expressing Kel-13 and the one is expressing GFP. So if you treat with heme, you see the intensity of the green fluorescence here. It's so high compared to absence of heme. Okay? But this is seen only in when Kel-13 is present. When GFP is present, you see the green intensity has barely gone up. Okay? So now what is Kel-13 doing? Heme generates reactive oxygen species. But you need 100 micromolar heme to generate reactive oxygen species. If Kel-13 is there, 5 micromolar heme is sufficient to generate reactive oxygen species. So that's what Kel-13 does. It generates reactive oxygen species, okay, so that at a lower heme concentration. Okay? So in fact, it stimulates 4 to 10 times more reactive oxygen species as compared to only heme. A Kel-13 bound heme generates ROS much more efficiently than just free heme. Okay? That is the function of Kel-13, to generate reactive oxygen species when heme is coming. Okay? What is happening to NRF2? If Kel-13 releases NRF2, what is NRF2? It's a transcription factor. Under stress, what would the transcription factor do?
It will come to localize into the nucleus.
It will go to in the nucleus because it needs to express stress response genes. There is no heme here, no stress. The red signal is cytoplasmic. Okay? But in case of heme, where is the red signal? In the nucleus. And that is when you see in the Kel-13 cells only. In the GFP cells, there is no translocation to the nucleus. It appears like this only. So Kel-13 cells, when heme comes in, it generates reactive oxygen species. As a result, NRF2 goes to the nucleus, starts expressing the stress response genes. So the stress response genes will start expressing. Now this is a Western blot that shows that in presence of heme, NRF2 goes to the nucleus. Okay? In absence of heme, NRF2 is in the cytoplasm. But in presence of heme, NRF2 goes to the nucleus. If it goes to the nucleus, what does it do? It expresses stress response genes. You look at the gray curves here. In presence of heme, it expresses stress response genes. So that's what KELS13 functions. Okay? So in terms of a model, if you see, you have to, at the end you have to visualize what is happening. Okay. Now, this rainbow color propeller is the KELS13. Okay? So KELS13 binds NRF2 and degrades NRF2. Okay? When heme comes in, heme is the red circle, solid circle. When heme comes in, it binds KELS13, NRF2 is released, which goes to the nucleus. It expresses stress response genes. Okay? So, what is heme doing? Heme is giving a pro-survival response. As soon as heme comes, release NRF2, express stress response genes so that you can survive, the cells can survive. So it's basically a pro-survival mechanism that the heme binding to KELS13 does. Releases NRF2, goes to the nucleus, and then it survives. Okay? Fine. This is the mechanism. The question is, how is KELS13 degraded? Heme provides stability of KELS13, but if there is no heme, how is KELS13? You saw that in absence of heme, the fluorescence intensity was lower. How is it degraded? How is the K13 degraded? There comes a mechanism which is called as autophagy. Are you familiar with autophagy? Autophagy is what? Eat me. Basically, when the cell starts digesting itself so as to survive the stress. There are different arms of autophagy. You have heard macroautophagy, microautophagy, where the cells also start degrading mitochondria, nucleus, other vesicles, it starts degrading. One of the specialized arm is called as chaperone-mediated autophagy. Have anyone read about it? It's very specific arm. What happens in chaperone-mediated autophagy is there is a protein called as HSC70. It's a heat shock protein. HSC70 recognizes the proteins to be degraded with a sequence called as KFERQ. Okay, so this is HSC70. There is a protein to be degraded which has a sequence called as KFERQ. Right? So HSC70 binds this protein, KFERQ containing protein, takes it to the lysosome. Right? The lysosome has a marker protein called as LAMP2. Every organelle has a marker. What is a marker for mitochondria? Cytochrome. Yes, right. Mitochondria is also powerhouse of the cell, so ATP synthase complex is there. Cytochrome C is also there. Okay, so lysosome has a marker called as LAMP2. Now, HSC70 binds the protein to be degraded. That protein has KFERQ sequence. It brings this complex to the lysosome, and then this protein is trafficked in the lysosome. The lysosome has cathepsins, acidic enzymes, which degrade the protein. This is what is called as chaperone-mediated autophagy. This is a chaperone, okay? This recognizes specific sequence which takes it to the lysosome for degradation. Now, is K13 degraded by chaperone-mediated autophagy? What do we need for that? We need this KFERQ sequence. So if you look at KELS13 sequence, there are 19 KFERQ-like motifs present in KELS13. Okay? These are present in different regions. And if you look at the images, what you can see here is, in absence of heme, low KELS13, and the red one is LAMP2A marker for lysosome. Okay? What is happening is the intensity of the KELS13 is low. It is at the lysosome for degradation. But if you have heme, it is still associating with lysosome, but the green intensity signal is quite high. Okay? Which means that it is here, sure, but not going here. Because had it gone here, it would have been degraded. The green intensity would be lower, but it is not entering the lysosome. It is there associating with lysosomal structures, but because it has heme bound to it, it is not entering the lysosome. So that's how it is surviving. So heme binding is preventing chaperone-mediated autophagy degradation of KELS13. Okay? Are we clear? KELS13 is degraded by chaperone-mediated autophagy. We'll finish it soon, don't worry. How do you check it? You can check it if you take care of LAMP2A. It is LAMP2A that is basically trafficking it inside. What happens if you lower LAMP2A? There's no trafficking inside. Okay? And the protein will survive. So, there is an inhibitor called as ATRA. ATRA inhibits LAMP2A. Okay? ATRA causes lower LAMP2A level. It inhibits LAMP2A transcription. So, now, you remember under no heme condition, there is low signal, low KELS13 because it is degraded. But if you treat with ATRA, ATRA will lower LAMP2, prevent degradation. Okay? So that's exactly what is happening. Under low heme, if you treat with ATRA, the green fluorescent intensity is increasing. So it's no longer getting degraded. In fact, it is very similar to the heme condition that is preventing the degradation of KELS13. Heme binding will take it to lysosome, prevent degradation. If you deplete LAMP2A, it will also prevent degradation. So KELS13 is degraded by chaperone-mediated autophagy, and you can actually detect that in Western blot. If you treat with ATRA, basically, you see the signal is higher here compared to the heme depleted condition where there is, where there is no ATRA. Okay. This is a bit complex. You have to, think over a little bit. K13 is like KEAP1. K13 stability is dependent on heme. KEAP1 is not dependent on heme. KEAP1 doesn't bind heme, not dependent. So what you do is, this protein binds heme, stability is dependent on heme. This protein does not bind heme, stability is not dependent. But alignment-wise, they are similar. So how do you check which region is conferring the heme binding stability factor? You basically do this chimera, domain swap. You take this domain from here, put it here, this domain from here, put it here. So that's exactly what we have done. This is KELS13, these are the propeller region. This is KEAP1, the purple or pink is the propeller region. So we made a KELS13 with a propeller region of keep 1, and we made a keep 1 with a propeller region of KELS13. Okay? So if you see in the images, you see only when the propeller region is of KELS13, it is showing the heme-dependent stability factor. Okay? The propeller region of keep 1 does not show the heme-dependent factor. In fact, that's what you can see. If the propeller region of KELS13 is there, it confers heme-dependent stability to keep 1, like in this construct. Sorry, in this construct, it confers heme-dependent stability, but not the other way around. If you change the propeller region of KELS13, put keep 1, it is not conferring the heme-dependent stability. Okay? Not only it does not confer heme-dependent stability, it also does not confer heme binding ability, in the sense that it is the propeller region of KELS13 that helps it bind heme. Keep 1 does not bind heme. Okay? And only the propeller region of KELS13 that is involved in heme binding. Don't give this too much complex thought. Just remember this point, that the propeller region of KELS13 is important. Artemisinin-resistant mutation are present in the propeller region. So that is how the parasite modulates artemisinin sensitivity by mutation in the propeller region, because it confers heme binding and heme dependent stability. Forget about this thing. Now, finally compiling everything, taking into this factor. Till now we talked about these two. What about this factor? What about the artemisinin factor? Previously it was said that lower KELS13, lower hemoglobin uptake, lower heme. If there is lower heme, lower activation of artemisinin, survival. Okay? So we looked into this aspect. Is heme altering the sensitivity towards artemisinin? Okay. Now, what you can see here is, focus on this graph. Okay. If there is KELS13, heme increases the sensitivity of artemisinin. GFP doesn't do, keep 1 doesn't do very efficiently. KELS13 does. This one is keep 1 with the propeller of KELS13. That one also does. So basically, it's a propeller region of KELS13 which has the heme binding ability, and when heme is given, it becomes more sensitive to artemisinin. Okay? So that is how it is important. Forget about this part. What can be said is here that as the level of KELS13 increases, it becomes more sensitive to artemisinin. So KELS13 level, the higher KELS13 level, the more is sensitive to artemisinin. That's how the parasite, decides. First thing, either reduce KELS13 level so that there is less heme, less activation. Okay? Or make mutation in KELS13 so that it binds less heme, less activation of artemisinin. That is how the KELS13 mechanism actually persists. Okay? So if you prevent the degradation of KELS13 by ATRA will inhibit this. So if you prevent KELS13 degradation, it will have more KELS13. Okay? So if it has more KELS13, it will be more sensitive to artemisinin. So overall, the mechanism is like that. We talked about this thing that in presence of heme, NRF2 is released. It will induce stress response gene expression. But when artemisinin comes in, this is KELS13 bound heme. When artemisinin comes in, what happens is this heme artemisinin, it will generate reactive oxygen species, it will cause death. So artemisinin basically stops the pro-survival mechanism, goes to pro-death mechanism. That is how KELS13 connects to heme and artemisinin. And that is how the mechanism of KELS13 mutation conferring artemisinin resistance is important. Okay, so I think we are here. Finally, this is the model. Now, if you have any doubts, just go ahead.
Sir, in slide number 13, you have mentioned that heme inhibits protease to digest K13.
Heme inhibits protease protection, yeah.
So, which protease you have used?
This is trypsin.
Trypsin. So, sir, trypsin generally cut at lysine and arginine.
Right.
So, can we say that heme just alter the structure of K13 and these lysine and arginine amino acid just put inside into the protein structure?
Good. Yes, that's exactly what we want to say. It changes the conformation of the protein so that lysine and arginine, which are previously exposed, now are hidden. So that's why trypsin cannot access. That's how it confers protection. Okay? Yeah, that's exactly the point. Yeah. Ago, nobody bothered to look into it. Yes, we just started with, and then we came up with, these all things. There are much more experiments here. Like 70% of the experiments I'm not shown because I just wanted to, convey that story without making it. As you will be eventually coming into research, you will find that the problem that you start to address, yes, you will be able to solve that problem, but it will generate 10 more questions. And those questions become much more interesting.
I have one more question.
Sure.
When K13 protein is present in cytosol, there are many types of protease like trypsin present in.
Yeah.
So when heme is not bind with the K13, how it is intact in the cytosol?
If heme is not bound to KELS13, then how it is protected? How it is protected? Now just consider in case of malaria. It's a bag of hemoglobin. Okay. The hemoglobin digestion occurs in the later stages where KEL13, okay, so in the initial stages the K13 level is low because there is less heme, the K13 is degraded. Okay, that's good enough because you don't need K13 function. In the later stages, because of hemoglobin digestion, there is so much heme, what you can say is K13 is saturated with heme. Then it is protected by, and remember one thing, trypsin, we use trypsin to check the digestion, okay? In the cell, okay, where is trypsin present? Trypsin is not activated, okay, it needs activation. Most of it is in the proenzyme state, so it needs activation. This is just to show that there is a conformation change, as you mentioned, which prevents accessibility of lysine and arginine that are essential for trypsin cleavage, okay?
And which is more efficient for the artemisinin resistance on the K13 or the homolog K13 KEAP1?
Keep one does not confer artemisinin resistance. Okay, so that's the thing. If you look at protein evolution, it starts with a common ancestor. Then gradually, as you evolve, there are changes in the protein which makes it acquire specific characteristic required for a particular organism. Okay, so that's how K13 KEAP1 evolved from a common ancestor. But now K13 is malaria line, KEAP1 is a human cell line. Okay, so as it evolved for malaria, it acquires new characteristics, which is different from KEAP1, the heme binding characteristic, the heme dependent stability characteristic, which KEAP1 doesn't have, because it did not need, because there is no, in mammalian cell and all, how much exposure to heme does it get? But in malaria, because it is in a bag of hemoglobin, there's a lot of heme. So that's why the amino acids got modified so that it can counter heme effect. Okay, that's evolution. That's what evolution is. Okay. Okay. Yep, I think we're good. If there is any question, anyway, what to say is the lab is at the first floor. Feel free to walk in. If you have any questions or you generally want to know what we are working on, etc., other than this, please come along. Okay? Thank you.